So James and myself, Gareth, we were assigned to look at the cardiac muscle cell. We wanted to look at the cardiac muscle cell, the difference that it has between itself and a skeletal cell. We also wanted to look at the shape of the cardiac cell, its location, its function, and also try and include some interesting facts and features about the cell. So the first thing we're going to look at is the difference between a cardiac cell and a skeletal cell. And the main one of that being is that the cardiac cell will contain a lot more mitochondria than the skeletal cell will. And this will usually be around 5,000 in a cardiac cell, whilst a skeletal cell will contain around 1 to 2,000. So the reason behind this is that a cardiac cell, because it contracts, will need a lot more ATP, a lot more energy. So hence the need for more mitochondria to create this demand for energy. So the next thing then we're going to look at is the location and shape of a cardiac muscle cell. So the cell is located in the walls of the heart. This can be seen just around here. So the cardiac cell will make up the majority of the heart's kind of wall and structure. There's another image showing the location. So the cardiac cell itself has a branch shape to it. So we can see that here, the way it has quite a thick trunk, and then these trunks will usually kind of diverge into two smaller branches if we go back to this one. So again, we have this thick trunk that diverges, and again, this thick trunk that diverges into these kind of branches shape. So this is how the cell gets its shape. The cell is striated and it has a single nucleus. So if we see these vertical lines here, these are the striations, and then again, these are the nucleuses. If we go to the images, these vertical lines here, these are the striations, and these are the nucleuses. So as we were saying, each cardiac muscle fiber contains a single nucleus and is striated or striped. This is because it appears to have a light and dark bands when seen through a microscope. So these light bands, these will be the striations. It is striated because of its sarcomere structure, and it will have involuntary fibers that are cross-linked to each other.
Next we looked at the functions of the cardiac cell. The working myocytes are specialized for contraction. They are found in the atria here, both atria and the ventricles. They are specialized muscle cells called cardiac myocytes. The contraction of the cardiac myocytes is initiated by electrical impulses known as action potentials. Unlike skeletal muscle, which have to be simulated by the nervous system, the heart generates its own electrical stimulation. The heart can keep beating even when it's taken out of the body. The nervous system can make the heartbeats go faster or slower, but cannot generate them. The impulses start from a small group of myocytes called the pacemaker cells, noted here. And you can also see them here. So they're almost the same as myocyte cell, but they're pacemaker cells. These constitute the cardiac conduction system. They are modified myocytes that will lose the ability to contract and become specialized initiating and conduction action potentials. The SA node is the primary pacemaker of the heart and initiates all the heartbeats and controls the heart rate. So the SA node starts up here, and then you can see it's like a stone being dropped into water that it just radiates a big circular pressure now. So it starts off small and then radiates outwards. If the SA node gets damaged, other parts of the conduction system may take over its role. This will be the AV node, or the bundle of his. The cells of the SA node fire spontaneously, generating action potentials that spread throughout the contractile myocytes of the atrium. The myocytes are connected by gap junctions, which form channels that allow ions to flow from one cell to another. So basically, these are the gap junctions there, and these are the ions. Some get lost in the actual action potential, and some of them going to go through. This enables electrical coupling of the neighboring cells, and the action potential in one cell triggers another action potential in its neighbor. And the signals propagate rapidly to the AV node and slow down a little to allow the atrial to contract, and then follow the conduction pathway and spread to the ventricular myocytes. So basically the SA node starts here, radiates out through, contracts the two atriums, and then there'd be a gap between here and here where it hits the AV node. And by the time it comes all the way down here and contracts the ventricles, these will be already in relaxation stage. Action potential generation and conduction central for all myocytes to act in synchrony, so it's very important that they all work together in synchrony. Pacemaker cells and contractile myocytes exhibit different forms of action potential. So next what we're going to do is we're going to look at the actual contraction potential itself. So we'll look for phase 4, 0, 1, 2 and 3, and then it goes back to phase 4 again. So this is basically the start and at the finish of the. So the phase 4 is the resting phase. The resting phase potential in the cardiomyocytes is minus 90 millivolts due to constant outward leak of potassium through the inward rectifier channels. So you can see the potassium continuously being leached and the sodium continuously coming in. So this the sodium coming in and potassium going out. So the Na plus and the K plus potassium and the sodium channels are closed at resting in the transmembrane potential, which will be minus 90. The action potential is triggered in a neighboring cardiomyocyte or pacemaker cell to cause the transmembrane potential, which we'll call the TMP for handiness sake, to rise above minus 90 millivolts. The fast Na channels start to open one by one, and the Na leaks into the cell, further raising the TMP. So the potassium leaks in at phase 1, or phase 0, depolarization. The TMP approaches minus 70 millivolts. The threshold potential in the cardiomyocytes, this is the point where enough fast sodium channels have opened to generate self-sustaining inward of sodium current. Their large Na current rapidly depolarizes the TMP from 0 to 0 millivolts or slightly above it. The calcium channels open while the TMP is greater than minus 40 and cause a small but steady influx of calcium down its concentration gradient. Phase 1, which would be just above here, so this is the TMP, and then this is just above 0, so come back down to 0 there. TMP is now slightly positive. Some K plus channels are open briefly, and outward of potassium runs the TMP approximately 0 millivolts, so 0 will be just above there, just above that too. Phase 2 is the plateau phase. Calcium channels are still open and there's a small insert, so constant inward current of calcium. This becomes significant in the excitation contraction coupling process. The potassium leaks down its concentration gradient through the delayed rectifier calcium channels. The two countercurrents are electrically balanced and the TMP is maintained at a plateau just below 0 millivolts through to phase 2. So that's phase 2 there, so that'll be the plateau phase, and then we're going to phase 3, which will be repolarization. So phase 0 is depolarization, and then we'll go back into repolarization. Calcium channels are gradually inactivated, so that means to stop the calcium coming through. The persistent outflow of potassium is now exceeding the calcium inflow and brings the TMP backwards. Resting potential of minus 90 prepares the cell for a new cycle of depolarization. So back here again, and then that'll be back to phase 4 again.
So the last thing we looked at then was just some interesting features and facts about the cell. The first is that we looked at is that the cardiac muscle cell is only found in the heart. This is the only organ in the body that it is found. Like we said before, it makes up the majority of the heart mass. So again, going back to the structure of the heart, the wall, the cardiac cell makes up the majority of that. The Purkinje fibers are large pale cells that are very rich in glycogen, but contain a very small amount of contractile filaments. Like we also said, the cell contains an abundant amount of mitochondria, so roughly around 5,000 per cell. And a unique feature that the cardiac muscle tissue has is autorhythmicity. So this means that the cell can set its own contraction rhythm due to the presence of pacemaker cells. So this is very unique to the cardiac cell. So thank you for listening.